Imagine, if you will, a world where we could rewrite the very building blocks of life, where we had the power to edit our genes just as easily as correcting a typo in a Word document. Would we seize the opportunity, or would we balk at the potential consequences? Welcome to the world of CRISPR-Cas9, a revolutionary development in genetic engineering that has sparked a global conversation about the ethical boundaries of scientific discovery. At its core, CRISPR-Cas9 is a method for altering the DNA of organisms, from bacteria to humans. It's a tool gleaned from nature itself, borrowed from a system that bacteria use to fend off viral invaders. This system, known as clustered regularly interspaced short palindromic repeats, or CRISPR for short, and the enzyme Cas9 form the basis of this revolutionary technology. So, how does it work? Imagine DNA as a long string of text in a book. Cas9 acts like a pair of molecular scissors, guided by a piece of RNA, a molecule that carries genetic instructions from DNA. This guide RNA is designed to match and bind to a specific sequence in the DNA. Once the Cas9 and guide RNA complex locates its target, the Cas9 cuts the DNA at the precise location. This break in the DNA strand prompts the cell to repair the damage. Scientists can exploit this repair process to introduce changes or edits to the genetic code. This, in essence, is the magic of CRISPR-Cas9. It's a technology that has transformed genetic engineering from a laborious, time-consuming process into something as simple as find and replace on your computer. So, what have we learned today? CRISPR-Cas9, a system borrowed from bacteria, is a revolutionary tool that allows scientists to alter the DNA of organisms. It works by using a guide RNA to direct the Cas9 enzyme to a specific sequence in the DNA, where it makes a cut. The cell's natural repair process can then be manipulated to introduce changes to the genetic code. The potential applications of this technology are staggering, from curing genetic diseases to improving crop yields. But just as breathtaking are the ethical questions it raises. Who gets to decide what constitutes a desirable trait? What are the long-term consequences of tinkering with our genetic code? These are questions we must grapple with as we stand on the cusp of a new era in genetic engineering. As we close, it's important to remember that while science often provides us with the tools to reshape our world, it is our responsibility to use those tools wisely. The story of CRISPR-Cas9 is just beginning, and it's a story that each one of us, as members of the human race, has a stake in. The future is in our hands, and with it, the potential to rewrite the story of life itself.